Good morning. Yes, I received your mails with the entire schedule of your lectures of the Georgia. Well, also got the email that they have been postponed till 9th of January, so we can continue at this time. Okay, so let's start with our today's session. Now, this is neuromuscular junction, but see, the thing is, it's not so that today we'll be restricting ourselves to only neuromuscular junction. We'll be coming across several fundamental things which will be the foundation for upcoming sessions. That is when we'll actually be going into the depth of the synaptic transmission. Once you know that, right, it would be very easy for you to understand so many processes, right? So today's session, not only the neuromuscular junction, but we'll also be talking about the spinal nerves, right? So there will be a bit of anatomy. We'll be talking about spinal nerves. What really happens when all those preganglionic fibers, postganglionic fibers, such terms when they come. So what exactly they mean? Technically speaking, we'll also be talking about reasonable depth, a medicine part. That is what is called as the upper motor neuron lesions and lower motor neuron lesions. Now, once you understand the concept, it will become completely easy. You don't have to really cram anything, right? You'll be able to figure out just by looking at the patient whether he is a case of UMN or LMN, that is upper motor neuron lesion or lower motor neuron lesion. We'll also be talking about that something when happens in the brain, what really would happen, that how the patient would represent. Okay. And yes, we'll be talking about obviously the neuromuscular junction. To start with, right? So let's start. So our topic for today, neuromuscular junction. Okay, neuromuscular junction. So here, what we are doing is we have got the neuron, and now instead of connecting it to next neuron, this time it is connecting to muscle. So here also this is a type of synapse only, correct? It is a type of synapse because at no point of time the nerve would be directly touching the skeletal muscle also, right? It won't, the way it is never touching the next neuron. Basically what it is releasing the neurotransmitter, right? So what we really understand that what are they doing, neurons, right? The electric signal which is coming, right? It is the electric signal, right? Electric signal. That electric signal is getting converted to chemical, right? It is converted to chemical signal in the form of neurotransmitter, right? It is the neurotransmitter. And that neurotransmitter, when it touches the next nerve, right, it gives the response. Now, whether it is stimulatory or it is inhibitory, well, it depends upon the neurotransmitter, right? That's what we understand. So, when this neurotransmitter acts on next neuron, that's how it works. So, can we really say that if everything in association or with respect to synapse, so we'll be having a presynaptic neuron and obviously the postsynaptic neuron. ठीक, right? So postsynaptic neuron. And this is the synapse where it would be the sort of neurotransmitter. Now that neurotransmitter will vary. This thing was discovered by Sharrington. Is related action potential part 1 and 2? Definitely. And that's why we'll be going back to even those notes also, because today I'll be adding something extra to that. Deliberately it was not said at that point of time just to avoid confusion, but today we'll be adding something extra to that, and that will make the picture so clear, right? This question which is asked, right? Do remember this name, Sherrington. Very important name because he is not considered as only the father of synaptic trans. He is considered as father of synaptic transmission, right? Synaptic transmission. But because this synaptic transmission is so vital, and that's why we'll be taking one special lecture only on synaptic transmission, right? Actually, what really happens at a granular level, right? Obviously that thing will be at such granular level, and at every point we'll say, okay, see, if the poison is given, this thing won't occur. If some drug is given, say this step would be broken, right? So that is where we'll be understanding it in still more depth. So this Sherrington, he is called as father of synaptic transmission, but he is also called as the father of neurophysiology, right? He is also called as the father of neurophysiology. And we'll really appreciate this thing, that why he's called as father of neurophysiology, when we'll be dealing with this synaptic transmission in depth. Right? Okay, so far it is synapse. But when we talk about a muscle, right? When we talk about a muscle and we say that there is a nerve, right? Here is a nerve, neuron, right? There is a neuron, and then there is a synapse, and this time there would be a muscle, right? So here is the muscle. So this synapse is a special synapse. Yes. I remember this. So that's why, Aishwarya, I told you that because in the benefit of everyone, I'll be talking about this calcium influx triggering contraction. Right? Today you'll be getting the answer of it. And that's what I said just before few minutes, that deliberately a small portion was not discussed. Say for example, in this was our note, right? When we talked about neuron, right, we said that okay, these are the potassium which will start influxing. This is our resting membrane potential, right? There is influx, that is there is this potassium, right? As these channels they open, right, gradually it starts rising, right? It starts rising, and then what really happens, it is the voltage gated sodium channels, right? They open, and then they lead to what is called as the sodium, right, voltage gated sodium channels, and that leads to depolarization. And at this height, these sodium channels, they are closed or the deactivated, and when they are deactivated, right, it is this potassium voltage gated channels open, and then there is repolarization and things continue. Well, there is one step which we have not discussed over here, that is the calcium part. That is what we'll talk about it. So we'll add something extra to this, and immediately the whole picture would be very clear. Okay. All right. So going back to our work. Yeah. So this synapse, right? This synapse. Huh. So this synapse, when we say about neuromuscular, so this is a sort of modified synapse, right? It is a sort of modified synapse, right? That means that there is a small difference between both of them. Now, one method is that straight away we go ahead with all these that this is what really happens, etc. Instead, what I thought is, let's look at the bigger picture, and then we'll keep on zooming all the way, so that immediately we'll know that this synapse, it is not so that it will be working on skeletal muscle. It will also be working on our organs, right? It will also be working on stomach and this and that everywhere. And when the terms which will be used, like means usually those terms, they are super confusing, that general visceral efferent preganglionic fibers and general visceral efferent postganglionic fibers, right? General somatic efferent, right? What really happens, how, why they are motor, what is dorsal horn, anterior horn, will make that thing super simplified, because it is necessary that when we'll be talking about sympathetic parasympathetic system, it should actually, you should be able to visualize that this is what is happening at every level. So that's why we are deviating from the topic, right? That's what I said. We are deviating from the topic, and then we'll come back to this topic so that we precisely know that this is what really happens. So we are integrating now the part of anatomy, part of physiology, and yes, definitely some medicine also will come into picture, right? And I know that when the medicine comes, you guys, you love it, right? And obviously it, we really understand that yes, this is why we should learn it. Because when I was in MBBS, always this question was that why we are learning this, what would really happen when we know this thing, but use kahan pe. The moment you know that yes, this is the use, yes, then it sticks to your brain. So let's talk about all those spinal nerves, right? Spinal nerves. Simple as that, that from brain, right, there is extension of spinal cord, you know, from there is extension of spinal cord all the way it comes in, and from spinal cord those spinal nerves they go out, and they go out to the entire body. Simple as that. If we divide the entire spinal cord, so then there is what? There is cervical, correct? Cervical. Then there is thoracic, right? Then there is lumbar, right? Lumbar back. Then there is sacral, and finally the coccygeal. These are the spinal nerves. So from cervical there would be 8 spinal nerves which will be coming out. Thoracic obviously 12, lumbar 5, sacral 5, and coccygeal 1. That gives us 31 pairs because they both emerge from both the sides, right and left. So there are 31 pairs of spinal nerves. Now, have you ever thought of, that's from spinal nerve, that is from spine, one nerve which goes out, right? It is carrying so many things. It is carrying few fibers for the muscles. It is carrying few fibers for the organs, right? So there is always a nerve which is carrying, it is carrying a bunch of fibers, right? And all those fibers, it is precisely like that your conduit. Conduit means that pipe, right? At your home, when you are wherever you are sitting, right? If you just look at, if it is concealed, then also it is okay. And if the piping, that electric pipe is out, you will find that one pipe, it is carrying one wire for the fan, one wire it is carrying for the light, one wire it is carrying for something else, right? Those plugs, one pipe, it is carrying all those things. Same thing is true for spinal nerve. So that's why we need to know very properly that how they are arranged, right? Let's draw, right? This is our spinal cord, right? And that's how it would be arranged. Spinal canal, and it has got the dorsal and the ventral side. Now these are called as the horns. So we'll call this as dorsal horn. Right? This one would be our dorsal horn. I am just writing D horn, and this would be the ventral horn. Correct? Now in between there is like a lateral horn also. Right? Lateral horn. But again, to avoid the confusion, right now we are not discussing lateral horn. Right? We'll discuss it when we'll actually be talking about those sympathetic, parasympathetic, the synaptic transmission in detail. At that point, we'll pick it up. So, but right now, dorsal and ventral horn. Remember one thing: incoming and outgoing. This is in, this is out. Whatever, whether the signal is coming from cutaneous, that is someone touches, or it is the acidity, whatever is happening, this is in, this is out. So when it is in, we call it sensory, right? Those sensations from where? From the entire body, right? So sensory, and this one is motor. Correct? It is motor. Now, at no point of time we are telling that whether it is under our control or not. No worries, right? We'll decipher it that whether it is under our control or not. But basically, if there is acidity, so yes, we are getting that sensation, and that has to be sent to brain. When we eat, our stomach is full. We don't tell our stomach, bas beta, now you are full, stop eating, right? No, it is an automatic process, right? That's okay. We override it, right? Especially when we go to marriage to vasulify that cover which we have done. So that is a different thing. Okay. So this is there. Now let's dive deep. So here is, once again, we draw right, and these are our horns. This is the central canal. Now when we say, let's draw, right? So here it is. This is like one nerve which is like a spinal nerve, right? And from here, and it would merge here, and there would be a ganglia, and it is going something like this. Looks confusing, right? What exactly are we drawing? Simple. Adding one by one. Things which are coming, we'll call them afferent, right? वो आ रही है, right? So this is R. So this is afferent. Things which are exiting, right? We'll call them efferent. ठीक? अच्छा. Whenever we come across something like this is ganglia. This is ganglion, right? No, there was no acidity. Was 142 gulab jamun, no acidity. I was absolutely fine. So ganglion. This ganglion this is our dorsal and this is our ventral. So we'll call this as the dorsal root and the ventral root. So that's why this ganglion is nothing but a relay center, right? A relay center. When the signals are getting weak, so this relay center will make it powerful and then send it further, right? So this is like a relay center at this point of time. We have to give some name to it. So we'll give the name as per its location. So let's give the name dorsal root ganglion. So simple, right? So far clear, dorsal root ganglion. Now the fibers, right? Now the fibers which are coming out from the exit point, that is from the ventral horn, right? From the ventral horn, the fibers which are coming out, they are carrying both the things. They will be carrying some fibers, they will be going to muscles so that they are under our control. Some fibers, they will be going towards the viscera, right? Organs, which are not in our control. So things which are not in our control, right? Remember, things which are not in our control, they will be like our autonomic nervous system, sympathetic and parasympathetic. True. So in this case, there would be few fibers, right? There are few fibers. They will come over here, and few fibers will be going directly. So fibers which are going to viscera, right? Fibers which are going to viscera, there would be two types, right? Viscera. And fibers which will be going to skeletal muscles. Fibers going to skeletal muscles, they will be called as the visceral, right? General visceral, right? General visceral. And those which will be going to muscles, right? They will be called as the general somatic. simple as that, general somatic. But one another interesting thing is that when it comes to, because over here also these are exiting, these are landing, these are the efferent, so we'll call that general visceral efferent. Similarly, general somatic efferent, because they are, whenever we say efferent, so that means they are coming from the ventral horn, right? So ventral horn is what over here only we saw, it is motor, right? So this motor part, that would always be efferent, and this sensory part, that would always be afferent. And this afferent, they are going via dorsal root ganglion. Good one. ठीक. So far, if it is clear, right, we add something more to this, and that thing will crystal clear every part. Changing the color, right? I need to have more colors, boss. Just a minute, right? Let's have one more color, and that would be this should be good. That's fine. Now see, this is we have not yet talked about this. This one is our beautiful sympathetic ganglion. Now sympathetic ganglion, listen very carefully, sympathetic ganglion is in the exit pathway, right? So here I'm drawing this, it comes here, and then the fibers are going out, right? So here, say this is like anterior horn. From anterior horn, fibers are coming, right? That's the sympathetic ganglion. And from here, the fibers, they are going to viscera, right? So these fibers, this is ganglion, right? This is sympathetic ganglion. So fibers which are before ganglion, right? Fibers which are before ganglion, we'll call them pre, and fibers which are after, we'll call them the post. Pre and post what? Pre-ganglionic fibers, right? Pre-ganglionic fibers. Good. So this would be, obviously we'll call that as post-ganglionic fibers. But which fibers? Right? These are sympathetic. Oh, sympathetic, that means it will be going to what? Viscera. So viscera, and it is from the exit point. So can we elaborate it in the form of that this is general visceral, right? Visceral efferent, it is going out from this anterior horn, right? It is going out from the anterior or the ventral horn. So general visceral efferent pre-ganglionic fibers, right? This is the full name, general visceral efferent pre-ganglionic fibers. Similarly, when we talk about these, so they will be what? Same. General visceral efferent postganglionic fibers, right? Rest is all same. So it is general visceral efferent postganglionic fibers. Perfectly fine, right? So this is pre and post. It is valid only for our viscera. That is our sympathetic system, right? Our sympathetic system. And this is exit, right? Exit. it is going out. So when it is going out, it means brain is telling something to the organs, you do this or you do that. That means these are all motor, right? These are all motor. They will send the message ki yes, you contract, or they say ki you don't contract, right? This means this is a motor system. Motor system, right? That is what is happening. What about when the viscera are sending the signals back? Wait, right? We'll talk about it, because they have to send it via the posterior horn, right? Because that is the sensory one. So we'll decipher everything separately and then we'll combine it. This part is good, right? Okay. Acha. Any doubt till this point of time? Any query? Because now we'll be taking a next concept. Any doubt till this point? We'll talk about all somatic, right? Afferent, everything. But any doubt till this point of time? If yes, do ask. If everything is good, we move on to the next concept. All good? Or any doubt? Even the slightest doubt, let me know. Let me know. Because once this concept is good now, you'll be the boss, right? In neurophysiology, all the confusions they occur because of these statements, these words. So any problem, any confusion, even if it is slightest, let me know. All good? Why it is not? Hmm. Is the chat box working? Okay. Chalo. So I take it like that, yes, this part is clear, right? No confusion. Let's move on to the skeletal part. General somatic efferent. In case of general somatic efferent, if you see, there is nothing like pre or post. That means it will be going directly. So what I mean to say is that when we talk about the skeletal muscle, as it will be exiting, it will be going all the way out, and it will be going to the muscles, right? Skeletal muscle. So that's why this is general somatic efferent. General somatic efferent, that would be motor, correct? Efferent is motor. General visceral efferent, we talked about it, pre and post ganglionic fibers, right? Now move on to the afferent one, right? In case of afferent, things are pretty straight. From the viscera, let's say this is stomach, right? So it would be general visceral afferent. General visceral afferent. It will be going, right? And then what about the muscles? For the muscles or the cutaneous sensations, cutaneous nerves, right? All those sensory things, they will be general somatic afferent, right? They will be general somatic afferent. So they will be sensory. They will be sensory, and they will also be joining. These fibers, right, collectively, they'll go all the way, and they will go into the posterior horn. Posterior horn, right? So thus, now we are say confident that what is general visceral afferent, what is general somatic afferent, what is general visceral efferent postganglionic fibers, what is general visceral efferent preganglionic fibers, and general somatic efferent, right? So pre and post ganglionic fibers, they are only for that autonomic nervous system, especially the efferent exit. Okay. Now, as we know that there are two systems, right? Sympathetic and parasympathetic. So draw it literally like this. So we have the top was cervical, thoracic, lumbar, and sacral. Here is parasympathetic. Here is parasympathetic. And here is sympathetic. Right? So that's why parasympathetic is also called as cervico and sacrum. So cervico sacral outflow. And for this sympathetic, it is called as the thoraco lumbar outflow. This is the reason that this is like abdomen, and in abdomen you'll find so many sympathetic ganglion, right? Those sympathetic ganglion, just because it is thoracolumbar, right? Thoracolumbar. In detail, yes, we'll watch, right, when we'll be talking about these systems. So again, this is a brain, right? From brain there is, so this is brain, then there is spinal cord, right? Then there is spinal cord, and from spinal cord those nerves, they will be going, these are the spinal nerves, and they will be going towards their target, right? Target. A very common word will come in the form of upper motor neuron lesions and lower motor neuron lesions, right? This thing invariably comes. This is UMN, upper motor neuron, and lower motor neuron. Why the chat box is so silent? Okay, let's hope that you are, all of you are listening carefully. So upper motor neuron, right? This is, it makes complete sense that when I write upper motor neuron means it is above anterior horn of spinal cord. Makes sense? See, above anterior horn of spinal cord. Is this good? Right? Because what was that anterior horn of spinal cord? That was our exit point of spinal nerves, right? So anything above this, right? Because from here, the whole thing in 3D, right? From here, the tracks will go towards the brain, right? So from here, right, they will go towards the brain up. So anything before this anterior horn is upper motor neuron, and after, it is called as the lower motor neuron, right? So from anterior, so below that, right, anterior horn of spinal cord, anything from anterior horn to the target is a lower motor neuron. And if that is cut, it is called as the upper motor neuron lesion. And if the lower motor area, any cut over here, it is called as the lower motor neuron lesion, right? It is called as the lesion, Lesion. Lesion means injury, right? Clinically speaking, we must know the difference between upper motor neuron lesion Yes, indirectly we have entered into neurophysiology, right? But then it will make rest of the things much easier. And this is lower motor neuron lesion. So everything fine till this point of time. Why there is not a single comment? Is the chat box working properly? Don't know. Okay. So upper motor neuron lesion, right? Where could it be? In the brain, right? There are tracks which are called as the pyramidal tracks, right? Pyramidal tracks. These are all higher level tracks or the extra pyramidal tracks. So extra pyramidal tracks, right? So any lesion over there, right? That is called as upper motor neuron lesion. When the spinal nerves are involved, so that is lower, because spinal nerves will always be emerging after the anterior horn. So spinal nerve lesions. So far this is okay, right? The second important point, any of the vascular accident in the form of hemorrhage, right, or the space occupying lesion, says in the brain if it is happening, so they are usually related to upper motor neuron. So it is called as the vascular accidents, right? Vascular accidents. We got new link of Zoom to join another lecture. Okay. Uh, okay, for your Georgian lecture. Chirag, is it for Georgian lecture? So vascular accidents and say they are called as SOL or the space occupying lesions. So something which is in the form of tumor. No problem, right? In any case, say this thing would be recorded, so you can just uh, go through it at your convenience, right? And whatever the queries, just put it into the comment box, right? So that say it is necessary. It is necessary, and do write something, right? Normally I don't say that do write your comments, but yes, it seems that it is important. So if you can write, it is well and good. Something, whatever, plus minus, you like it, something you don't like, any query, do write. Acha. Chalo. So no problems. You can join that Zoom lecture, right? So upper motor neuron lesion, when something is happening in the brain, right? So in the form of hemorrhage or tumor, and it will lead to lower motor neuron lesion will affect in which the spinal nerves are affected. Say for example, poliomyelitis, right? Poliomyelitis. So in which the spinal nerves, those lower end of nerves, they are involved. The third difference, which is so vital, and that is the tone of the muscle, tone of muscles. Right? Now let's write it this way: tone of the muscle and the power of the muscle, power of muscles. There is a very beautiful difference between both of them. So when there is no contraction, when the patient is at rest, right, you just squeeze the muscle, right, and that is the tone. Tone means tension in the muscle when muscle is at rest. So this we can write it like tension at rest, right? Tension obviously of muscle at rest. That is called as the tone, right? So this tone is increased. This tone is increased in case of upper motor neuron. While in case of lower motor neuron lesion, this tone is decreased. This tone is decreased. right? Tone of muscle is decreased, right? What exactly is power? Power of muscle is the ability to contract. Can this muscle contract, right? So that is contraction and the create resistance. Say this is contraction and create resistance. So here, will the muscle be able to show the resistance? Yes, it will be able to show, right? So this will be normal. This resistance will be normal. So at rest, if you see, the muscle will be quite tight, right? Quite tight. That tone would be increased. But in case of lower motor, it would be like jelly, right? It has lost its tone. What about the power? Power is lost. That means if you take the hand, right, it will fall, right? He won't be able to even generate. That is, the power is lost. Power of muscle, right? Power of muscle is lost. And this is the fourth major difference. Right? We are talking about upper motor neuron lesions and the lower motor neuron lesions. Upper motor means anything which is above the anterior horn, right? So due to this, right, how many differences we have seen? 4. Okay, let's have one more or few more, right? Here there would be paralysis because the nerve supply, when the nerve supply is cut, that muscle is paralyzed, right? So there would be paralysis. There would be paralysis. Here also and here also there would be paralysis. But see, this paralysis, if you have seen some patient, right, when they are paralyzed, so one thing is the hand is just hanging, right? And you lift it and then it falls. Lift it, then falls, right? There is no tension, there is no resistance, right? It is just loose. It is what is called as the flaccid paralysis. Flaccid means soft. It is the flaccid or the soft. Flaccid paralysis. Right? This is the case of lower motor neuron. While in case of upper motor neuron, if you see, right, so it's like if the hand has gone like this, right, so it will be so tight, right, it will be so tight, right. This is what is called as the spastic paralysis. Spasm, right, there would be spasm and it is called as the spastic paralysis, right. Clear difference, right. Spastic paralysis. Now see, when you'll be examining these patients, can you show location of anterior and posterior horn of spinal cord again? Why not? Why not? Right? See, this is where it is here. Right? This is posterior, right? Posterior horn, and this one is anterior horn. Anterior horn. Correct? This one is anterior horn. So that's why this is all going out. This is all efferent. Efferent means exit, right? This is where things are coming in. So this is afferent. Afferent means it is coming, right? It is coming. These nerves are रही है, right? So they are afferent. Everything is with respect to spinal cord, right? And these are the efferent. Efferent means these are exiting, and these exiting, they will be leading to, because they are going from the spinal cord, so obviously they will be, posterior is same as dorsal, exactly. So when things are going, so they will be giving some command, do this, do that, right? So that means they are all motor, right? They are motor. And the sensations which are carried by the entire body, and they are telling the spinal cord, okay, this is what is happening. So this means this will be sensory, whether the sensations they are coming from the cutaneous, from the skin, or from the organs, but whosoever, they are coming and going. Fine. Okay, so coming back to these lesions, right, coming back to these lesions, Posterior is same as dorsal. Now when you will be examining over here, now I'm showing you something because as such when you will be discussing the medicine, right? How to examine a neurological case, right? That you will be learning in medicine. But these small things, right, will make things much interesting. Here it is, right? Have you seen that Rampuri Chakku? Right, because there is no other name for it in English, so it has to be Rampuri Chakku, right? So they open that chakku, tick 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 thak, right? It opens like that. That initially there is resistance and then there is a spring, thak, chakku khul jata hai, right? So that's why those gundas, right? They speak the dialogue ke bas abhi dekh you, and then the chakku khul jata hai and then they say ke chal aaja, right? That's how it goes. This is what is called as the clasp knife, right? But Rampuri Chakku is always a good name. But we can't say Rampuri Chakku reflex, right? But do remember it like that. We can write it over here. But the name is clasp knife reflex. Reflex. So it is basically that Rampuri Chakku, right? So just imagine cut and thak, right? It opens. So it means that chakku, when it opens, right? That chakku, when it opens, right? From here, right? Initially there is resistance and then there is no resistance and it opens, right? This is called as the clasp knife reflex. Right? So here, this is the patient, and you just give the resistance, and it goes like this, right? And he is opening, and there is resistance, and then suddenly the hand is straight, right? Initial resistance, and then there is no resistance, right? That is clasp knife reflex. This thing is classically present in upper motor neuron. Here it is present. Makes sense because in case of lower motor neuron, as such there is no resistance. So what chakku kuch nahi, right? You just do this, it opens, you bend and it goes because muscles they are not giving, they are flaccid, they are not giving any resistance. So forget about this clasp knife, everything, right? There is complete absence. It is absent, right? It is absent. One another difference we see here, obviously the lesion is at a higher level, right? It is at a higher level. So many muscles would be involved. So here, group of muscles involved, involved, ah, involved, right? Group of muscles are involved. While in this case, usually single, right? Single muscle. Depending upon where exactly the lesion is, but usually single or smaller group, right? Or we can say smaller group. Smaller group is involved. One another interesting difference, right? If you see these patients, it is called as the clonus. Yeah, this is a very good difference, right? Clonus, very simple. What clonus happens? Muscle spasm, right? And then there would be some repeated rhythmic contraction, right? If you go to clinics, you'll find that some patient, right, there are muscles are quite tensed, right? And then there is continuous movement, right? Muscles, there is continuous rhythmic contraction. Clonus is that. So there is muscular spasm, right? There is muscular spasm. And when there is muscular spasm, it is leading to rhythmic, right? And see, when we'll be discussing the neurophysiology or the neuromedicine, at that point all these points they are having a specific reason that why it is happening, right? Because that control is lost. So that's why whatever the signals which are coming from the top, right, they have been inhibited. That means there is no inhibitor. So these are the things what are happening, right? So we'll see that thing in more detail. But this is rhythmic repeated contraction. Rhythmic repeated contraction. This is what is called as the clonus. Right? This is called clonus. In medical medicine book, you'll see that clonus is present, clonus is absent. But this is what is called as clonus. This is in capital. Yes, it has to be present because muscles, they are tense. Right? Muscles are tense. Tone is high. Power is high. So obviously they'll be giving this movement. No possibility of clonus in case of lower motor neuron lesion. You know, because muscles are flaccid, so they will be absent. Correct. So these are the differences which we see in cases of, say, both the things, right? Upper motor neuron lesion and lower motor neuron lesion. And it's not so that this list is totally complete, because there are some, say, Babinski sign which is present and which is absent. But right now we don't talk about it, right? Babinski sign is like on foot when you really scratch, so then all those flaring of the fingers, it is called as Babinski sign. Chalo, let's just write it, right? It is called as the Babinski's sign, right? Don't worry much about it, right? If you can remember the name, it is fair enough. If not, nothing to worry at this point. This is present. Babinski sign is present, and in this case, it is absent, right? To make things easier, you know the reflexes, right? You know the reflexes. When you tap, to then the hand moves, right? Say on the knee, if this is the knee, this is the knee, right? This is the knee, this is the foot, right? When you tap here, then you will get the movement of the leg. These are what's called as the reflexes. So these superficial and deep reflexes, right? There are superficial and deep reflexes. You will find that these reflexes, they are not present. So reflexes, superficial, right? Superficial and deep. These reflexes, they will be increased. In cases of upper motor neuron, right? While in case of, say, lower motor, because reflexes, they are always handled by the spinal cord. If the cut is there, so there is no point that any of the reflex would be there. So they are lost. They are totally lost in lower motor neuron lesion. So in fact, this is like a complete list we have discussed about upper motor neuron and lower motor neuron. Don't try to cram it, but try to understand it, right? It will make a big difference, right? Okay. So with this concept, now you see how easy it becomes. Coming back to our original one, right? When we said that minus 70 millivolt, that was what? That was our resting membrane potential, right? Resting membrane potential, right? And as it starts, right? So at one point, because this is where the sodium, those leaky channels, right, they start, and at minus 55 millivolt, these voltage gated sodium channels, they fire, right? So these are the voltage gated sodium channels, they fire, and they lead to depolarization, right? At this junction, these sodium channels, they close, and now they'll be opening off voltage gated potassium channels. They operate, and they will lead to all the way the repolarization, so much so that there is hyperpolarization, and then it will come back to normal, right? This is what we have already talked about. So here initially it was like potassium efflux, potassium leaving, potassium going, right, out, and this is the sodium influx, right? So minus 55, they trigger, and then the voltage sensitive sodium channels will open, large amount of sodium is coming, action potential fires, all good. This goes towards the positive part. This goes towards positive. Now see, at minus 40, right? This is what we have already seen over here also, right? Over here, right? This was our previous one, correct? Voltage gated channels, right? And that's how the neurons they were working, right? This we have already seen. Fine. Okay. So we just keep our this figure. We are adding something at this point, right? We are adding something at this point. That is that at minus 40, minus 55, and it goes to the at about say minus 40 millivolt, something happens, and that is where the voltage gated calcium channels open, right? Voltage gated calcium channels open. Now what calcium is doing, because see now this is the cell, sodium is already entering, right? Even the calcium enters, so yes, collectively they will go towards the dipole, right? Depolarization, correct? Collectively they are going, but those calcium channels, right? The calcium which is coming in, calcium is not much interested in the depolarization. That's okay, because of its ionic value, it will be helping. Most of the things they are done by sodium, but these calcium which has entered, they will do something miraculous. That's our axon, right? This is where, this is the end, right? These calcium, right? These calcium, when they reach over here, right? There are vesicles. Vesicle means these are like balls, right? These are small balls in which there is neurotransmitter. Let's say acetylcholine, right? Acetylcholine is there, right? So calcium will lead to exocytosis of these vesicles. Exocytosis, right? Coming back to this, what was that, right? So these are the vesicles or the vesicles, and they are having acetylcholine, right? That is the neurotransmitter. So neurotransmitter is into the form of balls. How it has formed into this vesicle, that is what we'll see into the synaptic transmission, that how this acetylcholine actually forms these balls and how they actually travel. But remember, one of the step, right? One of the step, this is like end of the neuron, right? This is like end of the neuron, right? And end of the neuron, here is the synapse, right? Here is the synapse, and here is the target, right? This is the target, and this target is sitting with all those receptors, right? Ke chalo, acetylcholine come, right, and will lead to some changes onto the opposite side. So this acetylcholine should be released. For that purpose, when these balls they go out, the process is called as the exocytosis. Exocytosis, right? This process, exocytosis means they go out. So this is exocytosis of acetylcholine, right? For this, calcium is needed, right? So calcium needed for this purpose. So it means if you block calcium, this acetylcholine will not go out. It will remain inside. So effect would be stopped. Correct? So the point is that for the exocytosis of neurotransmitter. Now I'm just writing neurotransmitter, right? Because it depends that what neurotransmitter will be released, but whatever, calcium is needed, right? Calcium is needed. So for this neurotransmitter, two things are needed. One is calcium. Calcium needed. Second, we are learning one new word. It's called as the snare protein. Snare protein, right? Snare protein. This is a very, very beautiful concept, snare protein. Remember one word, right? This is synaptobrevin, right? Synaptobrevin. So this synaptobrevin, no big science, right? Is very simple. See this snare, right? Let's decipher this snare, right? That what exactly is this snare protein? It is nothing but it is like a one word, right? It is called as the snap receptor. Snap receptor. It means this is a site which will be helping in releasing the neurotransmitter into the synaptic cleft, right? Into the synaptic cleft. This is the synapse, right? So this is the cleft, synaptic cleft, right? The space in which these neurotransmitters should be released. So we know that calcium is needed plus this snare protein is needed. What this snare will be doing? They attach to the surface and then they make the way, right? They are like the big boss and they say कि चलो रास्ता दो, let the acetylcholine go out, right? So in case if this snare protein is affected, yes, it will lead to say restriction of that particular release. So this is synaptobrevin. So what exactly are this? Snare means snap receptors. So what is this snap then? Right? This snap is soluble in SF, attached protein, right? Attached protein. So okay. So this is SNAP, right? No. Don't confuse upper motor neuron and lower motor neuron for upper and lower limb, right? It is for an entire body, wherever the muscles are, right? Wherever the muscles are, where the lesion is. Because then, when we'll be learning in more detail, right, we'll see that where the lesion goes, right? So, if the lesion is into the say for example if it is into the sacral region, so obviously the upper limb would be spared, right? Upper limb would be spared because upper limb is getting the fibers from higher side of the spinal cord, right? So yes, but you can say it like upper motor neuron and lower motor neuron is not only for upper and lower limb, it is for the entire body. It is a concept, right? It is for the entire body. So that's why whenever there are lesions, the differences which we saw, they were from the clinical point of view. Because we can't go and check the stomach, right? We can't go and check the stomach. We can't go and check the lungs, right? There would be effects, but because the limbs, back, abdomen, right, there are specific reflexes. Upper limb and lower limb, very easy to figure out the muscles and the reflexes. So that is where the clinically will be able to see the difference, right? Otherwise, think it like upper motor and lower motor. What we discussed is for the entire body, right? Coming back to this snap receptor, right? Snap receptor. So this is soluble NSF attached protein. All receptors are sort of protein only. So that's why this is a receptor which is having soluble NSF attached protein. So now this, what is this NSF, right? This NSF is N-ethyl melamide so N-ethyl melamide sensitive factor. This is sensitive factor. This we have broken into total granule thing. This is N-ethyl melamide sensitive factor. That's the meaning. And if we go into still more detail, right, you'll find that these snare proteins, and yes, when it will come to neurophysiology, actually we'll study that also, that they are two types. One which is into vesicle. Now that's what we talked about, right? Vesicle. So this is called as the VAMP. VAMP is very easy. Vesicle associated membrane protein. So easy, right? So this is the vesicle in which there is acetylcholine, right? And this vesicle, a protein, right? This is vesicle attached membrane protein, right? Membrane protein. Makes sense, right? Makes complete sense. And this particular protein is what I was talking about. That is what is called as the synaptobrevin, right? Synaptobrevin. This is the protein, right? And because we are talking about this vesicle, there is another one which is the T, that is the target, right? But right now, let's not discuss it. That's fine. We are dealing with the vesicle. So everything in nutshell, that when we say that this neurotransmitter should be released, we require two things. One, the calcium, and second is this snare receptor or the snare protein, right, which is on the surface, which is on this surface. And this is what will help us into the exocytosis. Exocytosis means releasing this acetylcholine into the synapse and let the things work, right? So these are the two things which are needed. Perfectly fine, right? Okay. So here we were that for the exocytosis, calcium and the snare. That is synaptobrevin, they are needed. From where this calcium is coming? This calcium is coming from that minus 40 millivolt, right? The voltage gated calcium channels. That's from where the things are coming, and that's the cell, and this is the entry of that calcium. True? Achha. What if now we talk about the abnormalities or the clinical things? Think about that those sodium channels, right? Sodium channels. Those sodium channels, they were voltage gated, right? Voltage gated. What if we stop these voltage gated channels? Because voltage gated channels, they were leading to result in acetylcholine release, and acetylcholine release was actually because of calcium, right? Because calcium voltage gated channels, they were getting triggered, and that was leading to all those acetylcholine release. ठीक? And that acetylcholine will lead to all the muscle contraction and all the processes, right? Muscle contraction, etc. What if we block this? We block this, right? Say for example, there is a poison, right? This is a poison that is called as the tetrodotoxin. So that is tetrado, it is tetra, right? Tetrodotoxin. This tetrodotoxin, it affects these voltage gated sodium channels. So it blocks. When this is blocked, it will lead to, say, this muscle contraction, right? They will be affected. So there will be no muscle contraction. right? And this thing leads to what's called as the respiratory paralysis. Because for respiration, we require the muscles, right? So this is the respiratory muscle paralysis. Paralysis. So here it is said that in spite of the presence of air, person dies of suffocation. Because it is the respiratory muscles which are needed, right? So that you take the air in. This is a fatal toxin, right? This one. This is a fatal poison, fatal toxin. And have you heard of puffer fish, right? This is a sort of fish, right? It is called as the puffer fish. In the sting of puffer fish, right, this toxin is there. So yes, people extract it and then it can be used for various purpose, but then respiratory muscle paralysis, when it occurs, it is definitely, it leads to what's called death, right? One more important thing, few say this is our presynaptic area, right? This is presynaptic. This one is postsynaptic. Postsynaptic. And in between is synapse. Now see those calcium channels, right? Those calcium channels which are opening at minus 40 millivolt, right? Because these are voltage gated. These are voltage gated. What if they are blocked, right? What if they are blocked? When they are blocked. So in this case, right, this is presynaptic block. So again, acetylcholine won't be released, right? This condition, this is called as the Lambert-Eaton myasthenia gravis, right? Myasthenia gravis. In short, LEMS. The point to remember is that it is presynaptic. Do remember, right? These are some common questions which are asked. So Lambert-Eaton myasthenia gravis is because of the presynaptic calcium blockers. Why it should happen? Because body develops antibodies. Why body should develop against own? Autoimmune disorder. So that's why it is the autoimmune disorder in which auto antibodies, they are created against the calcium channels, voltage gated calcium channels, or those channels, they are blocked or destroyed. When they are destroyed, we are not getting the sufficient calcium, so these vesicle, it won't go for the exocytosis, so that this acetylcholine is not released into the synapse, and it leads to what is called as the Lambert-Eaton myasthenia gravis or the myasthenia syndrome, right? S is for syndrome. Oh, okay, let's correct it, right? So that is called as the myasthenia syndrome. Syndrome, right? S. When it is postsynaptic, right? When it is postsynaptic. Now, in this postsynaptic, what would happen? Well, there would be the receptors, right? These are the receptors which are ready to receive, which are ready to receive this acetylcholine, right? And these receptors, they are nicotinic. There are two types, nicotinic and muscarinic receptors, right? What will they do? We'll see that thing when we'll be talking about sympathetic, parasympathetic, and this synaptic transmission in detail. But just remember, this is like a nicotinic receptors. Nicotinic receptors. When these receptors are damaged, again, why these receptors should be damaged? Same reason, auto antibodies, right? This is auto antibodies. So again, autoimmunity disorder. So when this is damaged, right, postsynaptic, so this is what leads to myasthenia gravis. Myasthenia gravis, right? So in both the cases, what is happening? In both cases, it is the muscle, right? Whether it is LAMS or it is myasthenia gravis. In both the cases, it is muscle weakness and there is paralysis, right? There is muscle weakness and paralysis, right? Crystal clear. So that's how things really work. Acha. One more very nice thing. There are, say, this is the synapse, right? This is the synapse. Chalo, everything is fine and the acetylcholine is released. Acetylcholine is released, right? But this acetylcholine, right, it has to be taken back, right? Because otherwise this acetylcholine will keep on working on and on and on, right? So it will lead to hyperaction. So that's why in our body there is always a, a system, a control mechanism that this acetylcholine is released, it starts acting, and immediately there is acetylcholine esterase. He comes into picture and he will deactivate this acetylcholine. Okay, okay, bus, your function is over, right? No more, stop. So that gives a nice control. Now, for some reason, what would happen if this inhibition of inhibition, that means if this acetylcholine is destroyed, right? If this acetylcholine is destroyed, when acetylcholine is destroyed, say for example, organophosphorus poisoning, right? Organophosphorus poisoning, right? All those cockroach killers, medicine, those dawai, etc. Organophosphorus poisoning. So it will destroy acetylcholine esterase. This is inhibited. So if this is inhibited, this acetylcholine will keep on working, right? There would, because acetylcholine esterase is destroyed, so now acetylcholine, it gets free hand, right? There is no one to stop him. So there would be hyperactivity of acetylcholine esterase, right? The result is acetylcholine esterase, right? This is stimulating which system? Sympathetic or parasympathetic? Parasympathetic, right? Parasympathetic. Parasympathetic means this is for rest and digest. So what would happen to pupils, right? Pupils. It is the sympathetic system in which they were wide pupil, right? Because we have to fight. In case of parasympathetic, it is rest and digest, so that pupil would be pinpoint pupil, classical thing, right? So that's why whenever you suspect organophosphorus, immediately, right, you open, right, put the torch and you'll find that small pupil, right? It is what is called as the pinpoint pupil. Pinpoint pupil. When you see this, you are sure that this poison has started working, right? And another thing what would happen is sympathetic, this parasympathetic is going into hyper. It means it is into digestive mode, right? So all secretions, they would increase, right? All secretions, they would increase, right? There are so many other things, but if you remember this much, right? Because this is crystal clear, because of that parasympathetic system in which acetylcholine is constantly working, because there is no one to stop him. Acetylcholine esterase was there to control, but because that poison has affected acetylcholine esterase, and it is leading to hyperactivity of acetylcholine, right? This is one. Then one more thing, a very dangerous poison, right? These are like, let's give the thing, poisons, right? This organophosphorus is one poison. Another very dangerous poison is what is called as the botulinum, right? Very poisonous, right? It is called as the botulinum. this is super toxic. Now, what it will do, all these are questions, right? All these are questions because they are very specific questions, right? That where do they affect? Botulinum toxin, it will affect snare protein, right? It will affect snare protein. Now see, when it is affecting snare protein, right? Snare protein, you now know the name, synaptobrevin. So it will destroy this, right? Destroyed. This is needed, na? This is needed for acetylcholine release, right? So now acetylcholine will not be released, right? Will not be released because in order to break that vesicle, in order to go for exocytosis, it was necessary that this vesicle should break in and it should release the acetylcholine. Now, because of botulinum, it has destroyed that surface protein. So now that vesicle won't be able to break. He won't be able to reach to that target and come out and release, right? So result, no acetylcholine release, right? No acetylcholine release. When no acetylcholine release, there is no muscle contraction, right? So this will lead to paralysis. But what type of paralysis? It would be flaccid paralysis because no muscular contraction, right? So that's the difference how the patient would really present, right? So whenever it comes to, say, toxicology is so interesting, right? But all you need to understand is that where exactly are they acting, right? Say for example, organophosphorus poisoning, or say this botulinum, or or let's say these autoantibodies when they are generating, right? What exactly are they doing? How the patient is presenting, right? So all, say for example, this tetrodotoxin, right? This tetrodotoxin, say it is affecting the respiratory muscles. So this is how everything is so amazingly interconnected, right? So that's the importance of calcium, right? That's the importance of calcium. And parallelly, along with the calcium, we also learned the importance of this snare protein, right? Say, I was told that your lectures has been postponed till November, till 9th of January. So I received several mails about your entire schedule. That is on Saturdays and Sundays, there are no lectures, right? Otherwise, almost every day, the lectures are from 11 to 12 and from 12:30 onwards, right? So obviously you won't be able to attend. It's fine that you see the recorded thing, but I really wish that if you are there in live, the advantage would be that in case of any query, you can ask immediately, right? Anyway, no worries, right? We can always solve any of the query at a later stage. So we stop at this point, right? And we meet tomorrow with the next topic, right? For today, again, I would say do revise it very properly, right? Do revise it very properly. Don't try to cram, but understand the concepts, right? It would make a huge difference, right? Thank you so much and see you tomorrow, right? Same time, and we'll come up with one more interesting topic. And yes, as you said even in your emails that yes, we'll incorporate clinical part every day, right? Yes, we'll include the clinical part every day. Surely it can be done and it will make the subject more interesting. Thank you so much and see you tomorrow. Bye bye.